Welcome, everyone. Welcome, everyone. Let's begin. I'd like to start with prayer. So I am looking for a volunteer. Is there someone that could help out? Oh, please.
Our Father in heaven, we are grateful to you this day for the opportunity that we have to come together to learn and develop our understanding of Iochem. Please bless us that as we study and review the materials, that we might recall pertinent information and be able to make connections that might help us solve these problems that we're working on. We are grateful for the knowledge and expertise of a professor, and please bless him. These things we pray in the name of our Son, Jesus Christ.
Amen. Thank you. So quiz 2 will be due Monday at 5 p.m. I extended that a little bit because it kind of got out late. We are drawing near to we're almost complete. We have almost completed the grading of quiz 1. Those will be returned. There is a display case in the Benson building in the main east-west hallway, just to the west of the north and south glass doors. There are these large glass doors. So it's on the north side of the hallway. There is a display case for our class, and there are alphabetized bins beneath this display case. The quizzes will be returned to those bins when they are done, and they will be placed in the box according to the last name of the first person that appears on the quiz. So hopefully you can remember who appeared first on the quiz. I am doing a review, sort of an online review primarily today, shortly after class, about 3:10. I sent out an email. I hope you all got it, but that is gives you access to the meeting this afternoon. And let's see, anything else? Do you have questions about administrative matters or other things? Yes.
Will the review be recorded?
I have not found a way to record it on my on the version of Zoom that I have on my computer in my office. I will try, but right now I'm not sure. On this computer, it's an option right on the menu. On my computer, when I open up Zoom, there is no such option, but I will look and see if there's a way of doing that if possible. I'd be happy to record it and post it. But all right, let's begin. We need to finish some stuff today. At the end of last class, we had been talking about the Bohr effect. The Bohr effect is a reflection, or it talks about this relationship between pH and oxygen release. The more acidic the environment, the more oxygen that is released. And so prior to that, we talked about the partial pressure of oxygen as being a way of increasing oxygen release from oxygen bound to hemoglobin. So this effect of acidity is a second way in which oxygen can be released from the hemoglobin molecule. I'm going to spend a lot of time on the board today, but I do want to show you this slide to give you a sense of the extent of this effect. So let me bring this up. All right, so as you can see, on the bottom, we have partial pressures of oxygen. The partial pressure in the lungs is somewhere between 12 and 14, so it's not even on this particular plot. The partial pressure in the peripheral tissues is often somewhere between 2 and 4. It really depends on how rapidly, how extensively oxygen is being utilized by that particular tissue. So if you're talking about a very active tissue, then we may have partial pressures that fall as low as 2 millimeters of mercury of oxygen present. But you can see that there is a very pronounced effect of pH on this process as well. So let me just so if we look at the situation, if we have a partial pressure, find this, of 3, and we sort of come up from here, you'll notice that if we have a relatively small basic nature to our blood, that we're getting off somewhere. So if we start at 95%, we're down to about 55%. So that's a 40% of the oxygen is available under those circumstances. Notice that if we go all the way down to a pH of 7.2, which could be easily achieved in something like skeletal muscle or cardiac muscle, that we're going to get another 25% of the oxygen off. So now the delivery is about 70 to 75% of the oxygen picked up in the lungs is released now in the very vascular bed, in the very tissue where it is needed. And so the pH in the blood is not absolutely constant. It really depends on where in the circulation you are. So 7.6 might be exactly what you have in the lungs, but if you're climbing the hill, your skeletal muscle may have a pH of 7.2. So, and again, this drop in pH is caused by carbon dioxide being converted into carbonic acid. Carbon dioxide is a reflection of how active a particular tissue is. The more it's respiring, the more metabolically active it is, the more mechanically active it is, the more CO2 is being produced. That leads to increased diffusion of CO2 into the red blood cell. It's converted into carbonic acid, and it lowers the pH. Okay, so are there questions about this? This is called the Bohr effect. Anybody online have questions? Okay, let me take us back now to the board. And let me raise this a little. Okay. All right. I'm going to now talk about a second effect of carbon dioxide. So carbon dioxide is produced by all living tissues, by all cells. And it's able to, because of its being nonpolar, it diffuses out of cells into the circulation, into red blood cells. They're converted by this enzyme, carbonic anhydrase, to carbonic acid. That is one fate of carbon dioxide. So as the carbon dioxide comes in, we have carbonic anhydrase, this enzyme, which is going to convert it into carbonic acid. But there is a second fate for carbon dioxide. And that is that each copy of hemoglobin, so each hemoglobin protein has an N-terminus. Okay, this conceptually is the first amino acid in that entire polypeptide chain. This primary amine can interact with carbon dioxide in a reversible fashion to produce this species. This is called carbamate. Carbamate. So as CO2 increases, so let's say we have an abundance of this in our skeletal muscle as we climb up the hill to the Benson building or to the Marb, we're going to generate more of this carbamate. Now, the significance of this in terms of oxygen binding affinity is as follows. This negatively charged N-terminus can now participate, because of its negative charge, can now participate in an ion bridge between two copies of hemoglobin along that interface that slides, okay? So B2A1 or B1A2, these interfaces, this is going to interact across there and produce an ion bridge if the tetramer is in the T conformation. Put another way, the presence of this carbamate stabilizes the T conformation. It doesn't place it permanently in the T conformation. It simply causes it to spend more time in the T conformation, and the consequence of that is more oxygen is released. Remember, the T conformation, the T conformer is the low oxygen binding affinity. So as we weaken binding affinity, more oxygen is released. So this, you'll recall, led to increased acidity. Increased acidity caused more oxygen to be released. Now we have a second mechanism involving CO2 that leads to the stabilization or increased stability of the T conformation with increased oxygen release. Okay, any questions about this? Yes?
So the second method with the carbamate, is it also, because there's protons as a product as well, so does it add to the...
Yeah, this also adds to the Bohr effect. Okay, so we not only get production of protons from carbonic acid, but the formation of the carbamate also leads to increased acidity and increased oxygen release. Okay, good question. Let me point out that about somewhere between about 80 to 85% of the carbon dioxide goes to form the carbonic acid and bicarbonate. Oops, that's a 3. About 15 to 20% of the carbon dioxide binds to the N-termini of the hemoglobin proteins, and they're carried to the lungs. And as we start to blow off carbon dioxide, as we reduce its concentration, then this whole process shifts in the opposite direction. We now go back to the free N-terminus of each copy of the hemoglobin tetramer. Okay, questions about carbamate? Yes?
Just repeat that last portion, 15 to 20% of what?
Carbon dioxide is bound to hemoglobin protein carried to the lungs where it is released. So these are equilibria. So as you lose carbon dioxide, as you breathe it out in the atmosphere, this process is reversed.
Does carbon dioxide bind to all 4 subunits of hemoglobin, the N-termini?
It can. It depends on the concentration. So If you have really substantial amounts of CO2, you'll have more of the carbamate formed, and this will reinforce this conformational favoring of the T conformation. Yes?
Only the bottom one is reversible if you get rid of CO2. The top.
No, this one is also reversible as well. So, yeah. All right. Let me explain another way in which we get oxygen off of hemoglobin. Okay. So let's say that this is a red blood cell. Okay. And as mentioned, our carbon dioxide diffuses passively through the cell membrane. There it is converted into carbonic acid and on into bicarbonate. You'll notice that bicarbonate is negatively charged and will not diffuse across the cell membrane. So CO2, as it increases, it passively moves across the membrane, producing more of the carbonic acid, also produces more of the carbamate, but we're looking now at this formation of bicarbonate. Bicarbonate actually is prevented from accumulating substantially inside the red blood cell because there is a protein complex in the membrane, or several copies of it, in the cell membrane of red blood cells that allows for bicarbonate to exit the cell as chloride is brought into the cell. This is called an antiporter. It's actually named the chloride bicarbonate exchanger. So in this case, then, we have bicarbonate leaving the cell as chloride enters the cell. What is the significance of this? Well, chloride can participate in an ion bridge. It's a three-way ion bridge. So by three-way, I mean we have these have side groups that have positive charge on them. They're going to interact with this negatively charged chloride and form this three-way ion bridge if the tetramer is in the T conformation. So this three-way bridge is not available in the R conformation, but is in the T. So as chloride concentration increases, it tends to stabilize yet further the T conformation, allowing for more oxygen to be released. So again, more bicarbonate is a function of how much CO2 is being produced and released from metabolically, mechanically active cells. But another mechanism then for increasing oxygen release is this chloride, this increase in chloride concentration within the red blood cell. Okay. Questions about this? It turns out that bicarbonate is the primary buffer in our blood. So carbonic acid, bicarbonate in the blood provides the buffering system for the circulation. And the pH there is about 7.4, 7.5, depending on obviously where you are within the circulation within the body. As mentioned, in a rapidly respiring tissue, it could be as low as 7.2. We have about 25 to 28 millimolar bicarbonate in the blood normally. And if you lose that, if you have a lot of acidic species being produced, you can overwhelm the buffering system and the pH can drop quite substantially as you have acidic species. Yes?
So the hydronium ion doesn't stabilize in C conformation, so is it favorable for the cell to bring in chlorine to then stabilize in C conformation until it can get oxygen to bind, more oxygen? Does that make sense when I say, like, is it more favorable for the cell to get rid of the bicarbonate and bring in chlorine just to stabilize it slowly in the meantime until it gets more oxygen?
I don't think I'm quite understanding your question. So all of these are happening simultaneously. It's not that chloride comes, the bicarbonate as we produce bicarbonate, we increase the Bohr effect immediately. We also convert carbon dioxide to the carbamate, which also stabilizes the T conformation. Chloride also stabilizes or can stabilize the T conformation somewhat as its concentration increases. So all of the reduction in partial pressure also, because the oxygen binding is in equilibrium, it can also bring oxygen off of the hemoglobin. So all of these mechanisms increase the unloading of oxygen from hemoglobin, feeding those tissues that need it in real time, in the right place. So, yes?
I guess two kind of questions.
Yeah.
The T conformation, can it have like one oxygen on it or two oxygen on it? Does it have to have no oxygen on it? Or is there like a mix?
Okay, so let's just go back. So when we're talking about X-ray structures, so the solved three-dimensional structures of crystalline deoxyhemoglobin or crystalline oxyhemoglobin, they're Perutz, the original, the scientist who originally did this, only observed two conformations, T and R. But we know immediately as we look at the Hill plot that there is this region of cooperativity where we actually have an increase in oxygen binding affinity as you load more oxygen onto the hemoglobin tetramer. In terms of getting things off, again, this only makes sense if you're really able to access both of these conformers in a continuous fashion. So it's constantly switching back and forth between T and R, and it's really what fraction of the time does it spend in the R versus what portion of the time does it spend in the T conformation, as to whether oxygen is being bound more tightly, as seen in the lungs, or more weakly, as seen in peripheral tissues. I don't think I'm quite answering your question. So,
Yeah, so when chlorine binds to the T conformer, does that mean it has zero oxygen on it?
no, we never get to zero oxygen. Okay.
Like for that specific hemoglobin molecule?
No, not even. When we talk about BPG, you'll understand that it's not looking at some. It's not just the distribution of tetramers. 5% of the tetramers are in the T, and 95% are in the R at the lungs. And then you switch over to all deoxyhemoglobin for 30% of the tetramers in the peripheral tissues, and 70% are still in the R. That doesn't work, especially for something like BPG, which we'll talk about next. Other questions? Did you have a second question? Okay. Okay, let me talk about one more way in which we are able to bring oxygen off. And I'm going to move back now to and show you the following. So this molecule, 2,3-bisphosphoglycerate, is made in red blood cells. Okay, there's not a lot of metabolism, not a lot of synthesis that happens in the red blood cell, but this is one of the molecules that can be produced in the red blood cell. Increases in BPG occur in oxygen depleted environments. So if you are at a higher altitude, if you are working in Mexico City at 8,000 feet, you will have higher BPG levels than somebody who's at sea level. If you want to, and the reason that this is important is that in the hemoglobin tetramer, when it's in the T conformation, there is a cavity at the center of the 4 copies of hemoglobin. So if you look, you can see that there is a small opening right here, okay, where this arrow points to BPG. This cavity, this pocket, is only available in the T conformation. When you're in the R conformation that's shown over here, as we talk about that rotation and these copies of hemoglobin sliding over one another, they pinch off that cavity. It is no longer available in the T conformation. So as a consequence, when we have higher BPG levels, again, we are reinforcing the T conformation. Okay? So this requires that this cavity be available for the BPG to bind. We'll talk about what's binding it in just a second. What this looks like in terms of the oxygen, fraction of oxygen bound, you can see that in the absence of BPG, so here we are out in the peripheral tissues between 2 and 4 millimeters of mercury, that without BPG, you cannot get oxygen off. Okay? In the presence of BPG, and you can see that there's 5 millimolar and 8 millimolar, small differences here, but in the presence of 5 millimolar, we're able to get about 40% more oxygen off the hemoglobin. If we look at 8 millimolar, which is not that much more concentrated, oops, here we are, we're getting somewhere like we're getting about 55% of the oxygen off. So a small increase in BPG results in more oxygen release. BPG is what distance athletes train to increase, okay? So if you are a distance runner, a distance swimmer, all of those long runs and long swims increase BPG. If you're going to climb Mount Everest, you don't try to make the entire ascent in one day. You go to a high elevation and you wait a day or two. So you start, let's say, at 14,000 feet, you wait a day or two, you have a camp there. This allows the BPG to be increased. Then you go to, let's say, 18,000 feet, and you spend another day or so, allowing the BPG to increase further. And then you would go to yet a higher elevation prior to actually making the final ascent. And even then, you would have oxygen present for the final ascent. What's happening is anytime you deplete the oxygen, there are transcription factors that lead to the increased production of BPG. So you had a question.
Would increased BPG make you get less oxygen like breathing in? Is that the higher elevation?
Well, look at this chart. So over here where we are, yeah, you're going to lose about 1%, maybe 2%. So instead of being at 98% saturation, you'll be at 96% saturation. But out in the peripheral tissues, you're gaining a lot more. You're gaining a lot more oxygen release in consequence of the BPG. So fortunately, because of the nature of this binding curve, we're able to get high levels of oxygen saturation even with lower BPG levels, or higher, even with higher BPG levels, I should say, but we get a lot more off in the end. Okay, let me move away from the slide here quickly. Where are we? Okay. All right. So let's look at the cavity. So we have now forming this cavity are 4 copies of hemoglobin. And if we're talking about now the beta hemoglobin, the beta hemoglobin presents 2 histidines and a lysine right on the edge of this cavity. Now, the lysine is probably positively charged. And these histidines are going to be variably charged. One of them is probably protonated most of the time. So the pKa, due to the microenvironment surrounding it, is going to be higher, such that at a pH of 7.3, 7.4 inside the red blood cell, that this histidine will be positively charged most of the time. This other one is likely to be uncharged or minimally charged most of the time. BPG has 5 negative charges associated with it. Each of the phosphates has 2 negative charges. The carboxylate adds a 5th negative charge. So 5 negative charges, and they're going to be attracted now to these positive charges, and the BPG is going to be electrostatically held in place. Notice that the Bohr effect is going to also influence BPG binding. So not only are we causing a salt bridge along this interface somewhere, but as we drop the pH, have greater acidity, we're going to cause the charge on these histidines to be greater, which will attract and hold the BPG longer, or for a larger fraction of the time, allowing the hemoglobin to stay longer in the T conformation, allowing for oxygen to come off. Okay. Questions? All right, let me just comment on fetal hemoglobin. So in the fetal hemoglobin, you have gamma hemoglobin as opposed to beta. And the significance of this is that you are missing the histidine that is positively charged most of the time. In other words, there are 2 fewer, or potentially 2 fewer positive charges surrounding this cavity. As a consequence, fetal hemoglobin doesn't bind BPG very well. And as a consequence of that, the fetal hemoglobin spends more time in the R conformation, which means that the oxygen binding affinity of fetal hemoglobin is going to be substantially better than that of maternal hemoglobin. The baby depends on oxygen from the mother to meet its needs. The placental membrane interfaces with a pool of blood in what's called the intervillous space, and the oxygen then can come off of mom's hemoglobin, move across the placental membrane, and bind to fetal hemoglobin. The fact that the fetus has higher binding affinity for oxygen allows for it to outcompete mom, and you shift the equilibrium to bring oxygen, more oxygen into the fetal circulation to allow the baby to get the oxygen that it needs. Okay? Yes?
At what point does fetal hemoglobin start to kind of transition into adult hemoglobin?
It happens at birth, and there are events that silence the gamma gene and activates the beta gene. I think the lifetime of red blood cells is about 120 days. So you'll start to see this happen. We'll still have residual fetal hemoglobin for a couple half-lives, but we'll start to populate the red blood cells in the baby, in the newborn, with beta hemoglobin. And so you'll start to transition to sort of the post-fetal hemoglobin, the alpha beta hemoglobin. Questions about this? Okay, I want to spend just a minute we talk about several different histidines in the context of hemoglobin. And I know it gets very confusing. It's hard to sort all of this out. So I'm going to kind of walk through this. Okay, and I'm going to, again, I'm going to really sort of focus on primarily this one copy of beta hemoglobin. Over here, we'll have the alpha, and here's the cavity, so there'll be another copy here and here. So somewhere here we have the heme pocket, okay? So let's go all the way back to the insertion of the heme group with the iron into the hemoglobin protein. And you'll recall that we specified two histidines there. One of them is the proximal histidine. I'm just going to indicate it with a P. And on the other side over here, there is this distal histidine, which is going to hydrogen bond with the oxygen, tend to stabilize it, tend to reduce its reactivity. All right? There's somewhere in here, and I don't know if it's on the alpha or beta, or maybe on both, there's a histidine that is going to become protonated as acidity drops and is going to form some sort of ion pair when we have this positively charged. That's a positive. When we have this positively charged, it will participate in an ion bridge provided the tetramer is in the T conformation. So this is the Bohr effect, okay? So the Bohr effect has nothing to do with the proximal or distal histidines. It has to do with a histidine that's on this interface that is sliding or able to slide. In one situation with a positively charged histidine, we can form an ion pair provided we're in the T conformation. And then there are these histidines down here which border the BPG binding cavity. So down here we have our BPG with 5 negative charges. We have these histidines here near the cavity that can be protonated and positively charged depending on the pH inside the cell. And also as a function of how much BPG there is. So if you think about it, BPG is binding to hemoglobin, and you then form this hemoglobin BPG complex. Oops. So if you increase BPG, you shift the equilibrium to favor BPG being in that pocket more of the time. When it's in the pocket, it has to be in the T conformation, but it's not permanently there. So, okay. A lot of stuff. Yes?
Yeah, sorry, can you say again what the histidine on the side, the top?
This is?
Yeah, what is, yeah, sorry, that one.
This one.
Yeah, which one, or what is it binding to, like creating that?
It is binding to a negatively charged species on the opposing copy of hemoglobin. Hang on just one sec. I think there's a slide that gives you a sense of this. If I can get there quickly. Oops, okay, let me go back. Okay, so here's one depiction of this. This is not a decided issue. So this is from one textbook. You can see the histidine 146 is the one involved in forming this ion bridge with a glutamate on the opposite side. Now, this only happens if the histidine is positively charged. So as you lower pH, you populate protons on that histidine. It then forms this ion bridge, which stabilizes to a greater degree the T conformation versus the R conformation. Our book shows it differently. It shows the histidine as being this HC3 histidine, and it's anyway, it's still interacting with a species on the other side over there, forming this ion bridge. So which histidines are responsible for the Bohr effect is still an open issue. It may be more than one. It's conceivable that you have pKa's that are both within range, and as you get to a pH of 7.2, that you have a fairly highly populated histidine or histidines on surfaces. The critical issue is that they form ion bridges with a species on this other copy of hemoglobin. That ion bridge is only available as you are in the T conformation and align the positive charge with the negative charge on the other copy of hemoglobin. Other question? Yes? Yeah.
Those are incorporated as well, is that right?
That is correct. One of them seems to be protonated more of the time. And I say that because in fetal hemoglobin, you're only but substituting another amino acid for one of these histidines. But that results in almost no BPG being bound. So that implies that this histidine, if you want to think of it, is not terribly protonated much of the time. But this histidine, which is lost in fetal hemoglobin, in the gamma hemoglobin, must be protonated most of the time. Otherwise, it wouldn't really make a difference. Yes?
Where's the lysine?
The lysine is right here at the pocket as well.
So is that also incorporated?
Probably, although, yeah. But again, it depends a little bit on the environment, okay? It's possible. So if you were talking about free lysine, absolutely. Be completely protonated all of the time. But in the context of a protein, we may weaken the basicity of that lysine, and maybe it's has a variable protonation state in hemoglobin. Yes? No, it's always happened. So the question is, do you have only the Bohr effect one time and only BPG another time? They're always happening together. So let me just say that the BPG is a slower process. So pH is immediate. It's the actual environment in which you find yourself. It's that tissue at that time. BPG, on the other hand, is an adaptation to your surroundings, if you will. If you have some sort of obstructive pulmonary disease and you're getting less oxygen, your body will try to respond by increasing BPG to allow more oxygen to get off. You're not getting enough, but maybe we can get more off and keep you able to move about and so forth, so you're not bedridden. It's as mentioned, it is what athletes try to achieve through strenuous workouts that actually make you somewhat hypoxic for long periods of time. Elevation, again, increases or reduced atmospheric oxygen increases BPG, but it's a transcriptional process that takes a day or two to be complete. Okay, we're really out of time for today. This sort of ends what will be on the exam. I didn't talk about sickle cell anemia. It's not on the exam, but it is an interesting problem. I recommend that you look at it in the book just for your own purposes. All right, let me end with a quick story. So I don't do a lot of things well, but I have two really substantial skills, okay, that I've developed over many years. One of these is washing dishes. I'm really quite good at washing dishes, okay? Nobody's ever gotten food poisoning in my household, so it's a big plus. The other one is folding and stacking chairs at church, okay? We lived in a ward back in Massachusetts where there was almost no tolerance. The stage, those little doors underneath the stage that allow the carts to go under with the folded chairs, if you had one sticking out an inch, you were in trouble. And so I became really sought after for ward events. If there was a dinner, if there was some sort of performance, they made sure that I was there at the end to make sure that the chairs were stacked just right so we could get them underneath the stage and put away. So I began to be in demand at ward events, and I began to notice some things. You know, it was interesting to me. We'd have a ward dinner, and about 15 minutes before the event was to conclude, there were those individuals who seemed to just evaporate with their kids, you know. About the time that people were starting to clear dishes and bang around in the kitchen to clean them, we would lose several families. They would seem to have this innate instinct to know that work was coming, and they vanished, okay. But there were those, I noticed, who were there at the end of every event. They were the same people. It was the same 10 or 12 people who were helping me put chairs away. They were the same 10 to 12 people who were washing the dishes, sweeping the floors. And it turns out that they were the same 10 or 12 people who were responsible for shoveling the walks of some poor sister in the ward who was a widow or who was a single sister. They were the same 10 to 12 people who were delivering lasagnas to some new mother, and they themselves had a van load of 5 or 6 kids hanging out the windows. I began to have this close association with these really remarkable people. They were the people who made the ward function. They were the people who went out of their way to be friends, to help, to do whatever needed to be done to make life easier for some other person, to make the ward function more effectively, more seamlessly. These were remarkable people, and I had the privilege of interacting with them simply because I could fold chairs and put them on a cart. But let me recommend to you that you stay a little longer at ward events, that you cultivate friendships with those people who are there, because they are people who are worth knowing and worth emulating. They are indeed disciples of the Savior. Thank you very much. Have a great weekend. Enjoy conference. I am going to do the review in about 20 minutes as soon as I can get the video compiled and on my thumb drive. Let's go back. Okay, so look at this. Okay, so under the most extreme conditions, lowest pH, lowest oxygen tension, you always have at least one oxygen.
There's more of a T conformer light.
Well, yeah. Here you're going to be spending a lot of time in there. Okay.
I just didn't know.
So, I mean, if you look at this, I mean, what happens at 7.6? You have 2 units of oxygen. There's no way, according to the roots, that you can be in the T conformation.
Why doesn't it know if it was like an average, like one has 4, one has 0?
Well, think about BPG. So how can it have an effect?
It has to be that slot.
It's in that slot. So if it can't get into the slot, it's not going to have an effect. So if everything's in the R conformation locked into it, BPG will have no effect. Okay. So if you're coming out of here with 95% saturation, everything's in the R conformation. There is no pocket for BPG. So how can BPG have an effect?
I just don't know if this is more of a law of averages.
Well, it is in a way, but you have to be able to access both conformers of hemoglobin to explain any of this.
I just want to make sure the Bohr effect, when we say, or we reference the Bohr effect, that's only referencing lowering the acidity of something, right? We
So
say
increasing the acidity, lowering the pH, and the exact mechanism, as I said, is still debated. Okay, so let's just go back here. We have this interaction between a histidine and, in such a way that it reinforces. So if you look at the second line here, this interaction only happens at a T conformation. So it tends to stabilize the T conformation,
okay.
allowing more oxygen to come off. So that's the simple explanation is I get more oxygen off because
That means different things.
yeah.
Okay, I just want to make sure, because, so the BPG and the chlorine help increase the Bohr effect? Is that a good way to say that?
No, I say those are more independent effects.
Okay.
So the formation of carbamate independently creates another ion bridge that stabilizes the T conformation. Now there is some acidity released as you form carbamate.
Okay.
So that contributes
Okay.
to the Bohr effect. Chloride is able to participate in its own ion bridge, a 3-way bridge that is, again, stabilizes the T conformation. So these are independent, but they're all happening simultaneously.
And they all just help to stabilize it.
Yes, you get oxygen off.
Awesome, thank you so much. Quick question, so these,
I think I am done. I have done all 4 parts. So you can take all of them.
okay, yes, I'm good.
And at some point, we need to sum up all of the individual parts.